is Mark, and today I will be discussing Python and R packages for interactive visualization of spatial single-cell omics data. These packages have been built around a tool that we recently developed called Vitessce, which is a visualization framework for multimodal single-cell omics data. The Vitessce framework was first implemented as a web application, as you can see in the screenshot on the right. But with these new Python and R packages, Vitessce can be used as a widget directly within Jupyter Notebooks, RStudio, or R Shiny apps. Vitessce visualizations are modular and configurable. As you can see in these screenshots, a configuration may include genome browser tracks, scatter plots, image and spatial data viewers, or controller components. As you may know, individual studies and consortia such as the NIH Human Biomolecular Atlas Program, or HuBMAP, are now routinely generating multimodal datasets comprising thousands of single cells profiled with assays such as RNA-seq, ATAC-seq, ChIP-seq, and fluorescence in situ hybridization. The outputs of these assays could include transcriptomics, epigenomics, chromatin accessibility data, or imaging and microscopy data. Computational analyses may also be performed to derive dimensionality reductions, cell segmentations, or cell clusterings and cell type annotations. There are currently many tools for visualization of individual single-cell modalities, such as genome browsers, image viewers, and scatterplot viewers, but there are currently few tools which enable visualization of all of these modalities simultaneously. Vitessce addresses this need as a visualization tool which can be configured to explore any subset of modalities profiled in a particular single-cell experiment. The need for Python and R package wrappers around Vitessce arose because as bioinformaticians, we typically access and analyze single-cell datasets using tools from the Python and R ecosystems. So it's most convenient to stay in the same environment when visualizing the data as well. The main features of these packages are functions for loading local datasets stored in commonly used single-cell data structures and file formats, functions for configuring the grid of visualization components, functions for coordinating parameters across multiple linked visualizations, interactive widgets for using Vitessce directly in Jupyter Notebooks, RStudio, and R Shiny apps, and functions for helping you to export datasets and visualizations to the cloud using object storage systems such as AWS S3. In Python, the package currently supports loading OME-TIFF images as well as ANData, Loom, and SnapTools objects. The R package currently supports loading OME-TIFF images and Seurat objects. However, we are actively working on functions to load data stored in other common single-cell file formats. To configure a Vitessce instance, first we need to import the package into our programming language environment. And next we add a data set and we attach an object containing our single cell data. Second, we configure the visualizations that we are interested in using. In this example, we add a scatter plot and a spatial plot based on the data set that we added in the first step. Finally, we arrange the plots that we defined in step 2 in a grid. In this case, we are using the hconcat horizontal concatenation function to arrange the plots in 2 columns. When we need to coordinate multiple visualizations, we can use the link views function. Here we specify that we want to coordinate the parameters of 2 plots corresponding to healthy and disease conditions. The resulting visualizations are linked on their zoom and pan interactions because the spatial target and zoom level were specified as the linked parameters. When the visualization configuration is ready, the final step is to create the widget. To do this, we run the .widget method on the Vitessce configuration object. Python and R packages also contain functions for exporting data to cloud object storage systems. Once data is in the cloud, a new configuration can be defined based on the remote files. This configuration object can be converted to a URL to save or share with collaborators. To learn more about the Vitessce, Python, and R packages, visit vitessce.io, where you can also find installation instructions and example notebooks. I'd like to thank the other members of the Vitessce team, as well as the entire Gellenborg lab and the HubMap data portal team. Thank you for listening.